Hello friends, welcome back to my channel Plant Pathology Pathshala. So in this video we will discuss about gene for gene concept or gene for gene hypothesis. So gene for gene concept was given by Flor 1946. Okay, remember both the scientist name as well as the year, both are important. Then he proposed this hypothesis while working on flax or linseed, that is Linum oxytaticum, and Melamsora lini, that is the linseed rust pathogen. So he discovered this, or he proposed this gene for gene hypothesis while working on flaxseed and rust pathosystem. Okay, flaxseed and rust, linseed rust. So what this concept explains? So this concept or this gene for gene hypothesis, it actually explains the host pathogen interaction at genomic level, at gene level, how the pathogen gene as well as the host gene interact, that ultimately result in disease as well as the resistance. Okay, so how this interaction occur, this is governed by gene for gene hypothesis given by Flor 1946. So the basic concept is, for every resistance gene in the host, there is a corresponding AVR or Avirulence gene is there in the pathogen, and vice versa. For every virulence gene in the pathogen, there is a corresponding susceptible gene in the host. So for every resistance gene, means where resistance gene will be present, definitely in a resistant cultivar resistance gene will be there. So if a plant is having a resistance gene, it is showing the resistance gene because the pathogen is having the avirulence gene. Without the avirulence gene, this R gene cannot express. Similarly, if a pathogen is showing virulence, means it is its ability to cause disease. It is able to cause disease. Why? Because the plant is carrying a susceptible gene. That's why the pathogen is virulent. Okay, so this is the basic concept, basic gene interaction at pathogen and host level, at genomic level, for which we are getting the ultimate result, either resistance or disease. So these are the genes. So 4 genes we have in case of plant. Either the resistance gene is present or absent. Absent means susceptible gene is there. Or in case of pathogen, either the virulent gene is there or avirulent gene is there. So 4 cases are there. Here either R gene is present or R gene is absent, AVR gene is present, or this small AVR means the virulent gene is there. So this, if you combine this interaction, then you will find that when, so first 2 cases R gene present means this is for the resistant cultivar, and when this R gene is absent means this is for the susceptible cultivar. So first 2 cases for resistant and second 2 cases for the susceptible cultivar. So 4 outcome we have. So 4 type of interaction and 4 types of outcome we will get. So when the host is resistant means R gene is present and the pathogen is also avirulent. Avirulent means pathogen is producing some kind of elicitor or some PAM molecule or something that can be recognized by the receptor of the host. R gene is nothing but just a proteinaceous receptor molecule that perceive or that recognize the pathogen and ultimately trigger the ETI response as well as other plant defense responses. Okay, so there must be a AVR gene present in the pathogen that will be recognized by our plant resistance gene. Then the recognition occur Due to the recognition, defense responses will be activated. So during AVR gene and R gene, the first response activated is hypersensitive response, and the disease, no disease will be there, and plant will show resistance. But when R gene is there, means a receptor is there in the plant, but the pathogen is not having any virulent gene, no elicitor is there. It is showing virulent. It is a virulent, it contains virulent gene. No AVR gene is there. Then the R gene is not able, receptor is there, but R gene is not able to recognize the avirulent gene, because avirulent gene is not there. So no defense activation is there, means even though the R gene is there, but it is unable to recognize the pathogen because of the absence of particular signature molecule. Okay, so ultimately the plant will become disease. It is attacked by the pathogen or the virulent pathogen. Then another case, in case of the susceptible host. So susceptible host means no R gene is there, no receptor or no recognition system is there. So whether the pathogen is virulent or avirulent, it doesn't matter. No recognition will occur. So no defense response. Ultimately, plant will become disease. So the disease resistance will develop only when there is a resistance gene is there in the pathogen, and a virulent gene must be there in the, a resistance gene is there in the host, and a virulent gene must be there in the pathogen. So during the interaction of R gene and a virulent gene, the recognition will ultimately trigger the defense response and plant will show resistance. Rest 3 cases, disease will develop. Okay. So this is the basic concept. Next, about the mechanism. Here the same thing. If the pathogen is avirulent, then avirulent means it will produce some elicitor molecule that is recognized by R gene, and ultimately the defense response will be triggered and plant will become resistant. But in case of virulent pathogen, no elicitor will be produced. Even though R gene is present or not, it doesn't matter. No defense, no recognition, no defense activation. Ultimately, plant will become diseased. So the classical examples of gene for gene hypothesis. So AVR9 and AVR9, this is for Cladosporium fulvum, the fungal pathogen of tomato. This is the AVR gene and this is the R gene. For tomato, then this AVR PTO, this is the avirulent gene, and PTO gene, this is the R gene in case of tomato. This is also a bacterial pathogen of tomato, Pseudomonas tomati, I think. Pseudomonas syringae pathober tomato, something like that actually. Check this. Again, the best example, most important, AVR peta. This is the AVR avirulent gene in case of Magnaporthe oryzae, that is recognized by the rice resistant gene peta, and this is for the rice blast pathogen. Okay. So the concept of gene for gene hypothesis is universal, but some deviations are there. So deviation are like, if a single R gene recognizes multiple AVR gene, then this gene for gene concept doesn't apply. Then the second case is, if a single R gene provides resistance for multiple races or multiple species of pathogen also, there also this gene for gene hypothesis fail. Another, the multiple allele for the R gene as well as AVR loci. Then more than 2 virulent genes may interact with the R gene. Then doses effect or addition effect may be there that reverse the dominance. So these are some deviation from the gene for gene hypothesis. Otherwise, this can be applicable for most of the pathogen or host interaction. So this is the basic about gene for gene concept. Its mechanism, examples and deviation. Remember the examples, okay? These are very, very important. Next, about the molecular basis of gene for gene relationship. See, we know that our gene and AVR gene are interacting, but how this ultimate result, either susceptibility or resistance, is developing, so that we will study in the molecular basis of gene for gene relationship. So the based on the molecular interaction, that is determining the host resistance or susceptibility, gene for gene hypothesis or relationship is classified into two type of interaction. Okay, so either it is incompatible reaction or a compatible reaction. So incompatible reaction will result in resistance, and compatible interaction will result in susceptibility. So first about the incompatible interaction or the resistance, how this resistance is developing. So incompatible reaction, this is seen in biotropic pathogen. Biotropic pathogen means those pathogen which need a living host for its survival or growth and development. It can't survive on dead decay organism. Okay, so it is an obligate pathogen. So incompatible reaction seen in biotropic pathogen. So here the R protein or R gene recognizes the AVR gene or AVR protein or the effector or you can say the elicitor, and this recognition will ultimately trigger the hypersensitive response, and hypersensitive response restrict the pathogen growth, and ultimately the plant will become resistant. See, hypersensitive response means this is a localized cell death just around the infection code. So if this is a leaf, at this region if a pathogen start infection, so hypersensitive reaction just means the cell death around the point of infection at the local level, so that the pathogen will not able to move to healthy cells. Okay, it will restrict the pathogen growth at that particular local level, and ultimately the defense responses will be further activated. So this is about the incompatible reaction in case of biotropic pathogen. But in case of compatible reaction, that is resulting in susceptibility, what happen? This actually occur in mostly in the necrotrophic or heterotrophic pathogen, means the pathogen that can grow on dead decay organism. So generally necrotrophic pathogen, what they do? So they actually produce some toxins first and then kill the plant in advance, then on the dead cells they will grow. So here already because at the initial stage only the plant is killed by the pathogen, so no recognition system, the plant is dead, so all proteins are already degraded, so no recognition will be there, so no effective recognition between the host and the pathogen, and defense responses are not activated, that's why it is showing susceptibility. So this is a compatible reaction for the pathogen. Okay, so pathogen will colonize and disease will develop. See, here in case of compatible reaction, inverse gene for gene hypothesis is applied. Inverse gene hypothesis means here the necrotropic effectors are produced, not the elicitor. Elicitor means compound that activate plant defense at very low concentration, but effectors are the compound that are produced by pathogen that modify the plant metabolism in favor of the pathogen and it trigger disease, but elicitor trigger plant defense response. This is the difference. I have already prepared a separate video on effectors as well as elicitor. You can check out that in the playlist. Okay, so here the interaction is between the virulent gene, virulent gene, okay, the small AVR gene, and between the susceptible gene. In case of incompatible reaction, the interaction is between R gene and avirulent gene, but here the interaction is between the virulent gene and susceptible gene, and this interaction induced means this recognition actually trigger disease. It will trigger necrosis, cell death, damage that benefit the necrotrophic pathogen. But in case of the incompatible reaction, this R protein and AVR protein interaction is resulting in hypersensitive reaction, that is resulting in ultimately the resistance. But here the interaction between the susceptible gene and the necrotrophic effector or the virulent gene leads to cell death or necrosis that promote the disease, no resistance. Okay, examples are the best example, this one in case of wheat, tox A. This is a susceptible gene present in the this is a AVR gene which actually interact with this susceptible gene, and ultimately necrosis is occurring. So this is about the basic interaction or molecular basis for gene relationship. So two major type of interaction occur: incompatible and compatible. Incompatible interaction for biotrophic pathogen, where R gene and the AVR gene interact, resulting in hypersensitive response. Ultimately resistance will develop, whereas in case of compatible reaction, susceptibility is induced in case of necrotrophic pathogen due to interaction between susceptible gene and the necrotrophic effector or the virulent gene. So this is all about the gene for gene hypothesis given by H.H. Flor 1946 while working in flaxseed and Melasora linea or linseed rust pathosystem. So this is very, very important. Remember each and every points and examples also. Okay, now I think it is clear to all of you. So thank you for watching this video.